Every year, 5.4 million Americans are diagnosed with skin cancer, and over 90% of those cases are caused by one thing: the sun. We accept UV exposure as a daily risk, but how does sunlight actually flip the switch that turns a healthy cell cancerous? Scientists at the University of Chicago have just uncovered a shocking cellular breakdown. It involves a single gatekeeper protein. When UV light destroys this protein, it unleashes a chain reaction of inflammation, paving the way for cancer. What is this protein, and is yours being weakened right now? We're breaking down the science you need to know. Stay informed with us at Senior Stop. We have a complicated relationship with the sun. It's the source of life, it feels good on our skin, and it's crucial for producing essential vitamin D. But this relationship has a dark side, one that we often underestimate until it's too late. We talk about sun damage, but what does that truly mean on a cellular level? For decades, the primary explanation has been straightforward. UV rays penetrate the skin and directly damage the DNA in our cells. This DNA damage creates mutations, and if the right or wrong genes are hit, a cell can begin to grow uncontrollably, leading to cancer. This explanation is true, but it's not the whole story. It doesn't fully explain the intense and immediate reaction our skin has to the sun, the redness, the heat, the swelling we call sunburn. This reaction is inflammation, and scientists have long suspected it plays a major role in cancer development, acting as a fertilizer for damaged cells. The missing link has always been the how. How exactly does UV light, a form of radiation, trigger this specific cancer-promoting inflammation? Now, a groundbreaking study published in the prestigious journal Nature Communications by researchers at the University of Chicago has finally provided the answer. They have uncovered a new layer of our body's defense system, a hidden surveillance mechanism that, when broken by UV light, unleashes the very inflammation that drives skin cancer. This discovery introduces us to a new crucial player in our cellular defense, a protein called YTHDF2. Think of YTHDF2 as a highly specialized security guard or a gatekeeper patrolling your skin cells. Its primary job is to maintain order and prevent false alarms. Specifically, it manages the cell's RNA, the messenger molecules that translate genetic information from DNA to proteins. YTHDF2 is a reader protein, meaning it identifies and binds to specific chemical tags on RNA, controlling their fate. In a healthy skin cell, YTHDF2 is abundant and active. It keeps a tight leash on all sorts of cellular processes, ensuring they run smoothly. One of its most critical and previously unknown jobs is to suppress inflammatory responses. It keeps the cell's fire alarms quiet unless there's a real emergency, like a viral infection. But the researchers at the University of Chicago found something alarming. When they exposed skin cells to ultraviolet radiation, the levels of this critical YTHDF2 protein plummeted. When we removed YTHDF2 from skin cells, we saw that UV-triggered inflammation was much worse, explains Dr. Yu-Ying He, the lead author of the study. This suggests that the YTHDF2 protein plays a key role in suppressing inflammatory responses. The sun, in effect, systematically disarms the cell's security guard, and when the guard is gone, chaos ensues. So what happens in the cell when YTHDF2 is destroyed by UV light? This is where the discovery becomes truly fascinating, revealing a precise, step-by-step molecular breakdown. It all revolves around a specific type of RNA that YTHDF2 controls. This isn't the famous messenger RNA you heard about with vaccines. It's a non-coding RNA known as U6 snRNA. Think of U6 as a set of sensitive instructions that, if left unattended, could be misinterpreted as a threat. Here is the normal, healthy process. 1. Tagging. The U6 snRNA molecule is marked with a chemical tag called M6A. 2. Binding. Our hero protein, YTHDF2, reads this tag and binds tightly to the U6 RNA. 3. Transport. YTHDF2 then escorts the U6 RNA to a specific compartment in the cell called an endosome. You can think of the endosome as the cell's recycling center. 4. Security. Once inside the endosome, YTHDF2's presence acts as a block, preventing the U6 RNA from interacting with anything else. It's safely sequestered. This process is a brilliant piece of biological engineering, a surveillance system designed to keep the U6 RNA from being seen by the cell's immune sensors.
Now, let's watch what happens when UV radiation hits that same cell. Guard down. The UV rays strike the cell, causing a massive decrease in YTHDF2 levels. The security guard is neutralized. RNA exposed. Without YTHDF2 to bind to it, the U6 snRNA is now free and exposed inside the endosome. Alarm triggered. The endosome also happens to contain one of the cell's most potent fire alarms, an immune sensor called toll-like receptor 3, or TLR3. TLR3's job is to scan for foreign RNA, like that from a virus, and trigger a massive inflammatory response to fight the infection. Mistaken identity. TLR3 cannot tell the difference between viral RNA and the now exposed self-U6 RNA. It sees the U6 RNA, mistakes it for an invader, and pulls the fire alarm. Inflammation cascade. TLR3 activates, unleashing a flood of inflammatory signals. This is the redness, pain, and swelling of a sunburn, but on a microscopic level. It's the cell screaming, "We are under attack!" This uncontrolled inflammation does two devastating things. First, it creates a hostile environment that can further damage DNA. Second, and more importantly, it creates a fertile soil that encourages mutated precancerous cells to grow and thrive. The very response that's meant to protect us is hijacked by UV light to promote cancer. As Dr. He puts it, our study uncovers a new layer of biological regulation, a surveillance system through YTHDF2 that helps protect the body from excessive inflammation and inflammatory damage. This isn't just a fascinating piece of science, it fundamentally changes how we should think about sun protection. It's not just about preventing DNA damage, it's about protecting our protectors. It's about preserving our YTHDF2 levels. Every time you step into the sun without protection, you are not only risking DNA damage, you are actively dismantling your cell's internal security system. You are telling your YTHDF2 gatekeepers to stand down, leaving the alarm bells wide open for inflammation to take over. This study moves beyond the simple "sunscreen stops burns" narrative. That burn is the problem. It is the physical manifestation of this YTHDF2 degradation and the resulting TLR3-driven inflammation. It's not a temporary inconvenience, it's a sign of a profound cellular defense failure. So what does this mean for the future of skin cancer prevention and treatment? It's incredibly hopeful. Before the study, the focus was primarily on repairing DNA or blocking inflammation after it had already started. Now, scientists have a new, upstream target. What if we could develop a treatment that protects YTHDF2 from UV degradation? Imagine a sunscreen of the future. You would still have the physical and chemical blockers, like zinc oxide or avobenzone, to stop UV rays from penetrating, but it might also include a novel compound that supports and stabilizes the YTHDF2 protein, ensuring that even the rays that get through can't disarm your cellular guards. Alternatively, for those at high risk, could we develop a lotion that disrupts the binding of U6 RNA to the TLR3 alarm bell? If you can't stop the RNA from being exposed, maybe you can just mute the alarm it triggers. This research opens the door to a new generation of targeted therapies that are less about brute force and more about re-establishing the cell's natural, healthy balance. By understanding this precise YTHDF2 U6 TLR3 pathway, we can intervene with pinpoint accuracy. But while we wait for these future treatments, the knowledge from this study makes our current prevention tools more critical than ever. The advice hasn't changed, but the why has become infinitely clearer and more urgent. Broad-spectrum sunscreen is non-negotiable. This is your first line of defense. You are not just blocking the rays that cause wrinkles, UVA, or burns, UVB. You are literally shielding your YTHDF2 protein from degradation. It's your external security force. Sun protective clothing, UPF. A hat, sunglasses, and long-sleeved shirts are not just for comfort, they are physical barriers that do the job your YTHDF2 protein would be doing. Give your gatekeeper a day off by not putting it in the line of fire. Seek shade, especially during peak hours, 10 a.m. to 4 p.m. The intensity of UV radiation is high enough to overwhelm your YTHDF2 defenses quickly. Seeking shade is a simple act of cellular kindness. No base tan. The idea of a healthy base tan is a dangerous myth. A tan is not a sign of health, it is a sign of injury. It is your skin's panicked response to UV assault, indicating that DNA damage has occurred and your YTHDF2 gatekeepers are already compromised. This University of Chicago study is a powerful reminder that our bodies are networks of intricate, elegant systems. For skin cancer, we now know it's a one-two punch. UV light causes DNA damage, the mutation, and also dismantles the security system, YTHDF2, that would normally suppress the inflammation that helps that mutation become a deadly cancer. The sun is not our enemy, but it demands our respect. And respecting it means understanding, on this new, deeper level, what it's actually doing to our cells. It's not just a burn, it's a critical defense failure. Protect your gatekeepers.